Hello everyone, in this video I am going to talk about the phase 2 reaction of the drug metabolism. This is one of the most important concepts of the drug pharmacokinetic and one of the basic parameters of the medicinal chemistry. So in this video lecture I will talk about the phase 2 reaction, their introduction, main overview and the different type of conjugation reaction which are occurring in the phase 2 like glucuronide conjugation, sulfate conjugation, amino acid conjugation and the acetylation and the glutathione conjugation. So let's talk about them one by one. Let's discuss the general overview of the drug metabolism. Drugs are xenobiotic molecules are highly lipophilic, non-polar in nature. So first they will go through the process of drug metabolism in order to get converted into polar, ionizable or easily excretable form. During phase 1 metabolism, which occurs mainly in the liver facilitated by the cytochrome P450 enzyme, the drug undergo through the different reaction like oxidation, reduction and hydrolysis reaction. The purpose of these reaction is to introduce a polar functional group in the chemical structure of drug molecule or to unmask the already present polar functional group which is previously covered by the non-polar functional group. So now the drug is converted into the polar or ionizable form, and now the drug have the two choices: either it will easily excreted out from the body if it is in much polar form, or if it is not in polar form, it will further undergo through a next step, that is the phase 2 conjugation or the conjugation reaction or the phase 2 metabolism reaction. That reactions are facilitated by the transferase enzyme. They are present mainly in the endoplasmic reticulum, liver cells and also in the intestinal walls. So during the phase 2 reactions, the drugs are now converted into the highly polar ionizable easily excretable species that can be excreted in the bile or urine. So the main purpose of the drug metabolism is to convert a highly lipophilic nonpolar drug molecule into the highly polar easily excretable species. The important point to mention here is it is not necessary for all the drug to go through the both phase 1 and phase 2 metabolism. There are many drugs which can only undergo through the process of only phase 1 and then easily excreted out. And there are also the classes of drug which can directly go through the process of phase 2 conjugation. And this is the general overview of phase 2 metabolism. You can see the drug endogenous molecule the drug are xenobiotic molecule which are highly lipophilic in nature. They went through the process of oxidation, reduction, hydrolysis, and after that a new functional group that is the polar functional group highly reactive like electrophile or nucleophile has been introduced. Here you can observe this is the ether and these are the carbonyl, or it could be the ester or the acid group. And also here you can observe the OH group, SH group, that is the thione, and the NH. These all are the polar functional group which are introduced after the phase 1 reaction. Now the drug is ready to go through the process of the phase 2, because the important point for the phase 2 reaction is there must be a polar functional group which is easily accessible in the structure of drug molecule. So now the drug will go through the process of phase 2. The phase 2 conjugation reaction is facilitated by the transferase enzyme. It could be the glutathione transferase, glucuronic acid transferase or sulfate transferase, depend on the type of conjugation. These conjugation reaction depend again on these type of different functional group, like glutathione conjugation can occur in the ether or the carbonyl group, and the sulfate, acetylation and the glucuronic acid can occur in all these type of the functional group like hydroxyl, phenolic, thione and the amino group. So we will discuss about one by one of them in our upcoming slides. Phase 2 reaction. Phase 2 reaction are followed by the phase 1 drug reactions, and the purpose of them is to convert the drug into the suitable polar hydrophilic drug molecule which are easily excretable. They are facilitated by the transferase enzyme, and in these reaction a polar functional group is introduced which are basically the endogenous moiety like glucuronic acid, glutathione, sulfate, glycine, depend on the type of the drug. These reactions are also called as the conjugation reaction because in these reactions the drug metabolite are conjugated or condensed or combined with these endogenous moieties. So now they will now after going through the conjugation, obviously the drug molecule will become larger in size and also strongly polar. Why polar? Because all these molecules are hydrophilic or polar in nature. So now they can be easily excreted out into the urine and bile depending on the type of drug. They are facilitated by transferase enzyme like glucuronosyl transferase, sulfonyl transferase and the glutathione transferase. Let's discuss about the glucuronic acid conjugation. Glucuronic acid conjugation is the type of conjugation of phase 2 reaction. In this glucuronide formation will take place, and this is the type 2 conjugation of the most of the drug molecule which are excreted into the urine or the bile. The step involved in the glucuronic acid conjugation is first the activation of the glucuronic acid takes place. The activation of glucuronic acid will occur, and then the condensation of the drug molecule with the activated glucuronic acid will occur, that is the UDP glucuronic acid. And this condensation or the conjugation is facilitated by the UDP glucuronosyl transferase enzyme. This glucuronosyl transferase enzyme is mainly located into the endoplasmic reticulum of the liver, epithelial cell of the intestine and the other extrahepatic site. So the important point to note here that the glucuronic acid conjugation can occur in both type of metabolism, the hepatic as well as the extrahepatic metabolism. The transporter, first of all, what happened, the transporter and the xenobiotic molecule which will travel from the cytosol into the endoplasmic reticulum, where the UDP glucuronosyl transferase enzyme is available. There the condensation or the conjugation will take place, and then now the glucuronide and drug metabolite condensed form or the more hydrophilic conjugate will travel from the endoplasmic reticulum into the cytosol. And now after this the drug metabolite have become the more hydrophilic because of the presence of this glucuronic acid carbohydrate moiety. And why it is more polar? Because now it has become more ionizable functional group after the introduction of this polar glucuronide moiety. Its pK is now 3 to 4 range. So after this, due to this reason, this is now poorly reabsorbed by the renal tubule, and it cannot go back into the renal tubule. So that's why it can be easily excreted out into the urine or in some cases into the bile, depend upon the type of the drug molecule. The example of the endogenous substances which can conjugate with the glucuronic acid include the steroids, bilirubin and the thyroxine. Mechanism of glucuronic acid is the same like the transport of. So this is the reaction of phase 2 glucuronic acid conjugation of the phenytoin, that is the CNS depressant drug. We know that sedative, and this is the phenytoin structure. First it will undergo through the process of phase 1 reaction, and which is facilitated by cytochrome enzyme, and after phase 1 reaction you may observe a new functional group hydroxyl has been introduced into the structure, that is the hydroxylation reaction. Here you may observe before phase 1 there is no any polar functional group, so now a new polar functional group has been introduced. This is the phase 1 metabolite. After this, now the drug will undergo through the phase 2 conjugation reaction, which is the glucuronic acid conjugation facilitated by glucuronosyl transferase enzyme, and now it will get converted into its O-glucuronide form. Here you may observe the glucuronic acid carbohydrate moiety has been introduced. This is a highly polar conjugate, and it can be easily excreted out. UGT family is the human Urinyl glucosyltransferase transferase family is divided mainly into the two groups UGT1 and the UGT2. Depend on the location, like UGT1 are primarily responsible for the glucuronic acid conjugation of bilirubin estradiol, which is the estrogen steroidal drug, and the other estrogenic steroid. And the A3 and the A4 family catalyze the conjugation of the drug which have the tertiary amine, and after their conjugation they will convert it into the quaternary glucuronide and hydroxylated xenobiotics. The B7 glucuronide family of the enzyme is the highest number of the is responsible for the conjugation of the highest number of drug molecule or the substrate, including the 3 and 6 glucuronide conjugation of morphine, because in morphine, if you remember, the conjugation reaction occur at the 3rd and the 6th site in their structure, and the 6 glucuronide conjugation of the codeine is also facilitated by the B7 family. Approximately about the 40% of the glucuronide conjugate are produced by the B7 family and 20% by the A4 family, which is conversion of the tertiary amine to quaternary glucuronide, and only 15% by the A1 family. These are the data from one of the published research papers. O, N and the S glucuronides. The xenobiotic are drug molecule which conjugate form the conjugate with the O, N and nitrogen, oxygen or sulfur glucuronide must have the alcohol or phenol groups in their structure, and they are from the ether glucuronide. The aromatic and some aliphatic carboxylic acid will form the ester glucuronide, or they are also known as the acyl glucuronide. Aromatic amine will form the N-glucuronide, because in aromatic amine the amine NH will react with the glucuronic acid and it will form the N-glucuronide. The sulfahydryl compound will form the S-glucuronide, where the conjugation of the glucuronide will occur with the sulfur, both of which are more labile to the acid as compared to the O-glucuronide. So they are the acid labile, so means they are the acid sensitive. Sulfate conjugation is a second type of conjugation reaction of the drug molecule. This occurs mainly in the steroid hormone catecholamine neurotransmitter, that is epinephrine, norepinephrine, thyroxine, bile products and the phenolic drug. The major physiological consequence of this conjugation reaction is now the aqueous solubility is increased and the drug is become more ionizable form and is easily to excrete out. The sulfate conjugation is ionized at the physiological pH, and due to this it has a smaller volume of distribution than the unconjugated steroid. So for example, if a drug molecule which is going through the sulfate conjugation, before conjugation its volume of distribution is larger, because it is still in the non-polar form. Once it will conjugate with the sulfur, once it conjugate with the sulfate, now it will become more ionizable form, and now its volume of distribution will be decreased. So but in some cases what happens, after the conjugation the drug activation will take place. That is, if you remember the concept of the prodrug molecule, in which the inactive drug will be converted into the active moiety. So in certain cases the sulfate conjugation can result in the bioactivation of the reactive electrophile or the therapeutic active conjugates. Like the example in this case is the minoxidil sulfate. In the minoxidil drug, the sulfate conjugation will take place, and after this conjugation the inactive drug molecule is converted into the active drug moiety. Conjugation with amino acid. This is the important route of metabolism for the xenobiotic drug molecule with the carboxylic acid before their elimination. It mainly occurs with the amino acid like glycine, the most important amino acid, which form the water-soluble ionizable conjugates. With which type of the drug molecule in which aromatic, aryl aliphatic or heterocyclic carboxylic acid is present. So in those drug molecules the glycine conjugation will take place, and now they are less toxic and their precursor acids easily excreted out into the urine or the bile. During this reaction, basically the amide bond formation will take place, because we know that all these groups have the carboxylic acid. Now these acids have to conjugate with the amino acid, which have the NH2 amino group. So the bond formation between the amino NH2 and the carboxylic acid will ultimately lead to the formation of the amide or the peptide bond between the xenobiotic carboxylic acid and the amino group with the glycine. Next reaction is the acetylation. Is also a type of phase 2 reaction which occurs with the transfer of the acetyl coenzyme, and mainly occur in the aliphatic or aromatic amine or amino acid, hydrazine or the sulfonamide group. The important point to remember here is the acetylation reaction occur in all those drugs which have the accessible amino group, like hydrazine, sulfonamide, or the amino aromatic amine or aliphatic amine. And the reaction which we discussed previously, that was a glycine, that is the itself is amino acid conjugation, but the drug molecule in which carboxylic acid group is present, in that molecule the glycine conjugation was occurring. So the sulfonamide drug, if you will go through my lecture of the sulfonamide medicinal chemistry, you will observe the different classes of the sulfonamide, N1 and N4 sulfonamides, and their SAR. You will observe that their conjugation or the metabolism will occur at the N1 or N4 sites, and these metabolic reactions are basically occurring by the help of this acetylation reaction. The secondary amines are basically not easily acetylated, so these acetylation reactions are mainly occur into the primary amine, and the acetylation produce the conjugation that retain basically the physiological activity of the parent drug molecule, like the N-acetyl procainamide is one of the conjugated drug example of the acetylation reaction, which is still active after the conjugation reaction. And some acetylation reaction are the slow and some are the fast acetylators. So the slow acetylation reaction will cause the accumulation of drug molecule into the blood plasma, and due to the accumulation in the higher concentration, the drug induced toxicity may occur in comparison to the fast acetylator. So the example of such drug in which the slow acetylation is occurring and the drug toxicity is occurring, the example are the hydralazine and the procainamide induce lupus erythematosus, and the isoniazid induce peripheral nerve damage, and the sulfasalazine, which is one of the sulfonamide drug, induced hematologic disorder. Isoniazid is, we know that the anti-TB drug. The lecture of isoniazid medicinal chemistry is also available. You may go through it. And the fast acetylation sometimes causes the conversion of drug to relatively non-toxic N-acetyl metabolite. So these are the difference between the slow acetylators and the fast acetylators. There's another important conjugation type, that is a glutathione conjugation and the mercapturic acid conjugation. Mercapturic acid are basically this S derivative of N-acetylcysteine, which is synthesized from the glutathione. Glutathione is basically generally accepted that the most drug metabolite through the mercapturic acid, and then they will go through the conjugation with the glutathione. This conjugation reaction is catalyzed by the glutathione ACE transferase enzyme. In human, there are two isoforms of the glutathione ACE transferase enzyme, that is the GST-M1 and GST-T1. The principal drug substrate enzyme is the GST-M1, and it is responsible for the conjugation of the drug having the nitrous or urea functional group, and like the example is the mustard urea type the drug molecule, which is the class of the anti-cancer drug, and that this type of glutathione conjugation reaction will occur. So this was all about the phase 2 reaction drug molecule metabolism, and now Let's check out the general overview of these reaction. Like this table is showing the all the different type of the conjugation reaction, and the conjugating agent with the agent or the enzyme, their conjugation occurring, the enzyme responsible for the conjugation, activated intermediate, and the functional group in which these conjugation reaction will occur. Like first, glucuronidation will occur. The conjugating agent will be the glucuronic acid. It will be activated by the UDP glucuronosyl transferase enzyme, and the activated intermediate we have discussed is the UDPGA. And these type of conjugation reaction will occur all those drug molecule in which hydroxyl carboxylic acid, NH2 amino or SH sulfahydryl group are present. The sulfate, amino acid and the glutathione conjugation will occur with the help of the sulfate, glycine and the glutathione conjugate molecule by the help of sulfotransferase, acyl transferase or glutathione transferase enzyme. And the activated intermediate are acyl coenzyme and the PAPS. So like we have discussed, the sulfate conjugation will occur in those in which hydroxyl and NH2 amino group are present. Acyl transferase or the amino acid glycine transferase will occur in the drug molecule having the carboxylic acid functional group and the NH2 group for the acyl conjugation. And the glutathione conjugation reaction will occur in all those drug molecules which have the alkyl halide, alkyl nitrite, epoxide and the lactone are having the mustard. Like we have discussed the example, acetylation conjugation reaction will be occur by the help of N-acetyl transferase enzyme, and N-acetyl coenzyme is the activated intermediate. It occurs mainly in the drug molecule which have the sulfonamide functional group, NH2 or the hydrazine functional group. Methylation is also one of the conjugation reaction type that occur with the methyl transferase enzyme, and S-adenosyl methionine is the activated intermediate. Hydroxyl, NH2 and sulfahydryl are the group. So for the exam point of view, for the GPT exam or the any commission type of exam, this table is important, that which type of reaction is occur responsible for the which functional group. And there is another table, this is also important, that this table illustrates the different example of drug molecule for the different type of reaction, like the glucuronic acid conjugation occurring in the chloramphenicol, morphine, paracetamol, salicylic acid and all these drug molecule. And same way sulfate conjugation is occurring in the paracetamol and salbutamol. Glycine or what we say the amino acid conjugation will take place in cholic acid, salicylic acid. Glutathione conjugation will occur in the paracetamol, ethyluronic acid. So this table is also important for the different type of test in the exam, pharmacist licensing exam and many other commission related exam, drug inspector or hospital pharmacist. In these exam, sometimes the example of drug is given and the question is asked about the type of the phase 2 conjugation, and sometime like the question will be like the glucuronide conjugation will occur in which of the following drug. Different example will be given and you have to choose one. So this was all about the phase 2 drug metabolism reaction or the conjugation reaction. Thank you.